Thank you, Chairman Professor Chandrasekharan, for his introduction. Also, I am thankful to the organizer. I am not a, I don't belong to Humboldt alumni. Still, the organizer has given me opportunity to share my research work with this intellectual alumni. So, as explained by our chairman, that I am really working on the interface of chemistry and biology, usually using nucleic acid as a research tools in the Institute of Genomics and Integrative Biology. So before starting my talk, let me thank my group member, those who work hard and allowed me to come here and to present my data. So especially I am thankful to Hemant, Devojit and Smita. So their work I am going to speak today. So before going into detail, let me explain a little bit what is microRNA. All of us, more or less, we know that from DNA we get RNA, then from RNA we get protein. So during this process, some of the part of RNA, they do not code protein. We call non-coding RNA, and that happens. There is a biological phenomena called splicing, and this splicing basically gives some portion of the RNA to convert a real RNA which produce protein and something which is non-coding. So this part of non-coding get little bit processed by different cellular mechanism and gives a particular structure, hairpin loop structure with some bulges here and there, and poly-A tail. It is known as pre-microRNA, primary-microRNA, and this happens within the nucleus, and there is a protein machinery which is known as DROSA, recognize this particular three-dimensional structure of RNA and clips it at this position and gives a very nice three-dimensional hairpin loop structure of RNA. As I explained, it happens in the nucleus. There is a protein machinery which is called exportins 5, takes this pre-microRNA outside of the nucleus, brings to them into cytoplasm. Then another protein machinery which is known as a dicer become more active to recognize very processed hairpin structure of RNA, clips at these two positions to give this kind of double-stranded duplex RNA along with dicer. Now, depending on the thermodynamical ability of hybridization of either of this site, we call 3' and 5', I am not boring with that, they get separated out, and one of the strand is selected by another protein machinery which is known as a risk factor. So other strand gets generally degraded. Now, in case of mRNA, this is mRNA which is basically gives a production of protein. There is a region we called is or known as a 3' UTR. If this mRNA is giving certain amount of protein production or concentration, say this is the bar graph schematically, and this microRNA, small microRNA, finds some complementarity within this 3' UTR of mRNA. What happens? It doesn't allow to process protein machinery to produce protein from this mRNA and reduce the expression of this protein. Now, you assume if this protein is essential, then there is a pathogenic situation because protein is not getting produced as it is required. It is already well established last decades. This microRNA has tremendous biological load. They are involved in brain development, neuron development, heart development, and so on. So any aberrant expression always will lead to certain kind of pathogenic state in cell. So taking an example of cancer, there are two types of microRNA. One is known as oncogenic microRNA, another is tumor suppression of microRNA. Without going to the detail, the upregulation of microRNA cause tumor formation, or downregulation of this type of microRNA, tumor suppressive microRNA, gives again tumor formation. So that means this microRNA pathways or oncogenic microRNA pathways or tumor suppressors microRNA pathway becoming target to control tumor formation. So what is the current tool? Current tool is that you have a microRNA along with agoprotein, which is shown here. You take complementarity of this small RNA, which is oligonucleotide. You can chemically modify it to increase their stability, because normal natural oligonucleotides are not stable in biological fluid, and you allow them into the cell so that they complement to each other, and as a result now this microRNA is not available to bind here, and as a result you can rescue the protein production from this mRNA. So this is the current tool. But as you understand here, the length of mRNA is much more larger than the microRNA. The degradation of this microRNA is very slow. So at any steady state condition, the concentration of small RNA is much higher than the concentration of the long RNA. So if you want to really suppress the function of this microRNA, you need to always or continuously give a constant dose to suppress the function which is shown by this microRNA. So that is the current tool which is widely expressed, used in literature. But what we thought, to overcome that constant dose for a longer time, we could develop a better tool which has much more longevity or better activity. So what is that? So we call it a DNA enzyme or ribozyme, which is already known. So you can invent this kind of artificial enzyme against the microRNA. So this is the microRNA sequence. You can use some kind of enzyme, nucleic acid enzyme, and in presence of some divalent cation, this enzyme is able to cleave the particular site of adjacent oligonucleotides, in this case microRNA. So the main problem here is that, so mechanism is that as it is an enzyme, it will bind and it will cleave, then enzyme will be regenerated and again it will bind and cleave. So this is a cycle process. The main problem is here is that this is very small, 20 to 22 mark in length. So if you hybridize them in a normal biological temperature, they are not stable enough. As a result, they do not bind very efficiently, so that the efficiency to cleave also very low. So why this is? As you know, the ribose of DNA or RNA bases, they have a two type of conformation, either S or N, and there is equilibrium, dynamic equilibrium between these two conformation. As a result, the base, when they are hybridizing, they sometimes open up and close. In case of knot conformation, base is very perpendicular and able to form a very nice hydrogen bond with the complementary base. So if we can lock this conformation and use a artificial basis to get the synthesize the oligonucleotide, then the dynamic equilibrium are prevented. As a result, you may get better stability. So there is a chemically engineered base which is invented by Jesper Wengel, and we are lucky to collaborate with him very early stage of this development to understand the biophysical aspect of this modified oligonucleotide, how they binds and what is the thermodynamical origin to give the extra stability and so on. So there are in numerous papers we have reported the rules and regulations how to use this locked nucleic acid in terms of their antisensibility. We have used this locked nucleic acid in our present case. What does do? So, if you have assume you have a double stranded oligonucleotide and the melting stability is that, you can assume anything. You replace one base by locked nucleic acid, say here T, the stability goes up. You can replace and replace, it will go up and then it will be saturated. So, 50% of the natural base can be substituted by this modified chemically engineered bases, and up to certain level like almost 10 to 15 degrees centigrade stability you can increase. So, we have used this locked nucleic acid placed in between this arm of this enzyme, and we have shown that the ability of the enzyme goes up drastically. So, there we have, this is a normal without the modification, this is 2 modification and 3 modification, and this is a quantitative measurement. So, this case you can clearly see that when we modify 4 bases, the efficiency of these locked nucleic acid enzymes goes very high with respect to normal case. So, we gave the terminology, it is antagomerzymes, and it was published in Anguinted Chemistry. Not only we have shown that in vitro it works, we have also went ahead, used the animal model. Zebrafish is a transparent fish which is basically vertebrate in nature, and very nice model system to establish whether this tool works in vivo or in a multicellular system or not. Again, this is nothing is there, no enzyme. This is standard enzyme which is used for zebrafish model, and this is the enzyme which we have invented, and clearly it is shown that the expression level of the target protein is going up when we are using the enzyme which we have invented. This was published in Chem Biochem. So, as far as what we have discussed, that microRNA is a therapeutic target, and we can invent new tools based on oligonucleotide. But oligonucleotide has an inherent problem because they are not as stable as small molecule that can be used as a drug molecule in principle, because of their several inabilities. They cannot be introduced into the cell very efficiently, as well as their instability into the cell. So, what is the alternative? Definitely, as a chemist, everybody will be convinced that small molecule is the only alternative. In principle, you can come out to design small molecule against any type of DNA structure or RNA structure if you know the structure, 3-dimensional structure. Pre-miRNA, as I explained you, it has very nice 3-dimensional structure with hairpin structure, some bulges here and there, which is unique for a unique miRNA. So, that means you can distinguish one miRNA to miRNA by small molecule. You can develop small molecule against any target, and this will bind anywhere here. You can design your molecule such a way that you can target them in a particular pocket, so that dicer is not able to clip anymore, so that you don't have this process further. As a result, you don't have microRNA in production. With this principle, we have taken several molecules little bit in a rational way. We have picked up initially the molecule which is known to target other RNA secondary structure, and these are the aminoglycosides. Like lot of you have heard streptomycin, neomycin, kanamycin, and so on. All these aminoglycosides are known to target, or their biological function are due to the targeting several different type of RNA secondary structures. So, our thought process was there to find out a scaffold, then modify, and then develop as a ligand molecule. So, we took 15 known FDA approved aminoglycosides and screened them against a microRNA which is well established to behave like a oncogenic. So, we measure the concentration of particular microRNA against the concentration of certain aminoglycoside, here in this case streptomycin. we established that this is a concentration dependent decrease of the microRNA. That means streptomycin may be binding to the pre-microRNA of miRNA such a way that it is inhibiting the production of microRNA. And this molecule we are getting from the screening, high throughput screening, which I am not explaining here. Sorry. As I told you also, if you suppress microRNA concentration, the target protein concentration should go up. Here the Western blot by which we measured the protein concentration, and you can see, this is a quantitative measurement. The concentration when we have larger quantity of small molecule, concentration of the target protein also goes up. We have done several other biological experiments to convince that it is real target. However, we took some computational followed by biophysical experiment to prove in vitro that it is real target and binding affinity it's considerably high. So we start with the computational study. We modeled them and try to understand their three-dimensional structure. This green color is the ligand and the blue and red patches are the RNA molecules. As you can see that the molecule are in depth with the minor group of the RNA molecule towards the loop size of the microRNA. We did some melting study. If you melt any kind of duplex DNA or RNA at a particular temperature, they will melt. Now if molecule is binding, the melting temperature goes up. So there we show that the melting temperature of pre-microRNA in presence of molecule goes up around 5 degrees centigrade, which is not great enough, but there is a reason why it is not great enough, and we will convince them that convince you that in a later few slides that it is a real binding. We did further analysis. We see that there is a nice hydrogen bond between ligand molecule and the RNA molecule, and we counted around 7 hydrogen bonds, which is good enough to target or to bind tightly. Then we try to understand where it is really binding. So far I do not know the molecule which we are talking about. There is a huge space of the RNA where it is exactly binding, though we have some idea from our computational study. So we want to complement our computational study by fruit printing assay. Here, for simplify for your knowledge, so we can understand by some cleavage. You have different kind of enzyme which can cleave any kind of secondary DNA or RNA structure. You can cleave them and try to understand which bases are protected or not protected against this enzyme. Now any molecule is binding to a particular pair of bases or region of bases, this will be protected against this enzyme. So 4-5 lane at the different concentration of streptomycin, and we see that this is the region, this is the quantitative data, this is the region which is not accessible for the enzyme. That means the molecule is going and binding there, and as a result it is not accessible for the enzyme. So we mapped back and understood that this is the region where the molecule is binding, and this is also little bit similar that we have, not exactly same, little bit similar, which we have obtained from our computational study. Now it is a huge molecule, it is not chemically synthesized this whole RNA. So what we have done, we have truncated, we have took this portion and get chemically synthesized by solid phase synthesis. It is commercially available, and we placed some fluorescent molecule in this region and tried to understand whether the molecule binding in this region or not. If it is binding, what is the strength of the binding? So we did again the melting study with this truncated version, and we see that there is a huge increment in the melting temperature. As I explained, the melting temperature increment is clearly indication that the molecule is binding. Then we did lot of fluorescence titration study. This is the isotherm. You can plot them and feed them with a particular equation, get the binding affinity. This is a molecule which we have obtained from our screen, and this is a negative molecule which was not positive to our screen. This is streptomycin, and this is something dihydrostreptomycin, similar to the molecule but negative, they didn't show. And we clearly see that there is a tight binding between the streptomycin and the pre-microRNA 21. So we reported again this work, it is anti-tuberculosis drug, it is well known drug that can be used or repositioned to target microRNA 21 and to inhibit its oncogenic function in nature. So during all this process, what we realized that molecules are synthesized by chemists. In test tube they will cleave. If cleaving is there, if you have microRNA and it is with your molecular beacon, it will bind, open up, you will see the fluorescence increment. Now any molecule which is interfering, it is basically is going to lead no fluorescence change in nature. So we did lot of study and showed that this process can be screened within 4 hours. We screened that, again established that this molecule, this process is fairly replicating with our in vivo screening system, and that was reported in SCS Chemical Biology last year. As I explained, so far I have shown that how to down-regulate microRNA. There are also a lot of microRNA which is suppressed in nature, you need to overexpress them. So as I explained, we are screening a lot of different type of molecules. This is another type of molecules are known to bind again RNA molecules. We screened them. This is global expression study. We measured all the microRNA which is available in cell and understood that this molecule are able to not target one, but it can target many, and when it is targeting, it is becoming better substrate, means my pre-microRNA is getting better substrate for the dicer. As a result, the concentration goes up. So we have proved different biological process. Yes, again the observation which we are getting from our whole microRNA genome-wide study is indeed by some molecular biological study. We proved that the few compounds are really able to rescue the function which is shown by tumor suppressive microRNA. Now there is a challenge. What is that challenge? MicroRNA comes in a cluster like a family, and 2 microRNA maybe only one base are different. Maybe C is substituted by T. So we call it as a microRNA family. And one microRNA is pathogenic, the other microRNA is not pathogenic. As I told you, they are very close to in sequence, it is very difficult to target again. But if you look at their 3-dimensional structure, they are completely different, which tells us that again you can develop small molecule to distinguish these 2 different kind of 3-dimensional structure. So we collaborated with Professor Tushar Chakraborty. He basically is very known to synthesize natural cationic cyclic peptide, and cationic cyclic peptides are known to distinguish very fine minute secondary structural change at the RNA level. We thought whether we can use this kind of cationic cyclic peptides for our purpose or not. Again he has given few 10 to 12 cationic cyclic peptide. We screened them against a particular microRNA 106A and B, and we have seen that yes, it is possible to distinguish 16A and 106A and 106B. So this work is under progress, because I have to again go back and check whether they are targeting the other similar kind of microRNA or not. So this work is going on. Then as far as I was describing, it is a basically screening. We need to come back and try to understand, design rationally. If I have given particular microRNA sequence, I should be able to design a small molecule without screening. So it is already known that this type of small molecule can distinguish a adenine, guanine, cytosine or uracil when they are not base paired with the counterpart. That means if you know this particular microRNA sequence, and if you know that which are the paired, which are not paired, then theoretically you can stitch them such a way that you can target maybe this region or maybe other region, whatever you would target for a particular microRNA. With this objective, we have started collaborating with Reddy from NCL and come out just 1 or 2 slides. Several molecules, screened them using our established tool, and found out one molecule is very effective. Again, this is just screening. We have to establish with complementary biological study whether this molecule are really a ligand for a particular microRNA 27A we are using in this case is real or not. So I have described a novel tool. It is a tool because, as I told, that it cannot be used for a therapeutic purpose. So we named as ontocoenzymes to silence very effectively, efficiently in vitro, in vivo microRNA function. We have also come out a nice methodology to screen small molecule in a large scale in test tube for microRNA. Also, we have established that small molecule can be designed to target or biogenesis of microRNA. And last, thank you all for your attention.
Yeah. And this, all the cases you are showing that as soon as this something is binding, the melting temperature going up.
Yeah.
Is that like
yeah, this is yeah.
It binding will give you its beta stability?
Yeah, most of the cases, when a small molecule binds to any secondary structure of DNA or RNA, it stabilizes. As a result,
I would like to take this opportunity to thank the organizers for giving me the opportunity to chair the session. Also thank all the speakers.
Wi-Fi, you will find something with net gear. This one, kind KINDFLUTE 06.
Who is a senior professor in the Department of Inorganic and Physical Chemistry. He earlier chaired the department. More importantly, he was an Alexander von Humboldt Fellow in the year 1994 in Ruhr University in Bochum with Professor Carl Wiegert, and he was also one of the founding members of the Humboldt Association in Bangalore. So he has several accolades to his credit. He has won several accolades. He is a Bhatnagar awardee. He was also the Third World Academy of Sciences Fellow and the Fellow of the 2 major academies in the country, the National Academy of Sciences in Bangalore and also in Delhi. And he is winner of the J.C. Bose Fellowship, and he has been working in the area of medicinal inorganic chemistry for a very long time, and he has published more than 230 papers, and he has to his credit, he has guided more than 20 PhD students. So with this short introduction, I invite him to chair the session.
Thank you, Professor Samuelson, for your kind introduction. I welcome you for this post-lunch session. We have 3 speakers in this particular, this is under chemical biology. So we have the first speaker, Dr. Suman Kumar Dhar. He did his graduation and post-graduation from Bardhaman University and Kalyani University. Then he did his PhD from molecular parasitology from JNU. And after that, he had a postdoctoral stint in the Harvard Medical School, working on DNA replication. Then in 2001, he joined JNU as faculty in Center for Molecular Medicine. And his area of expertise is parasite and bacterial DNA replication. All the 3 talks are of 30 minutes. I request the speakers to take 25 minutes and leave 5 minutes for discussion. So may I now invite Dr. Dhar to talk on this topic shown here. You can read that long title, In Search of Potential Inhibitors of Human Malaria Parasite, Plasmodium, falciparum in vitro and in vivo, a chemical biology approach. Thank you.
Thank you, Professor Chakraborty, for that introduction. Thank you, Dr. Mukesh, Dr. Devnath Pal, for the invitation, and also Humboldt Foundation, because my entry into the parasite field was initiated by Humboldt Foundation. Because I didn't know when I started working on Plasmodium, I never worked on Plasmodium, but Humboldt actually gave me the opportunity for a second postdoc after taking up this job. And I went to Hamburg, Bernard Nocht Institute of Tropical Medicine, and then there I got trained in parasite biology. So I hope I'll be able to keep you awake after this fantastic meal. And don't get afraid with this long title, because this chemical biology approach, when I got the invitation, I didn't know whether I fit in chemical biology. But just to make myself, happy, and I am talking about chemical biology, I put that. So for us, for biologists, what we do is we first try to understand the system, and then after you do that for 5 to 10 years, then the question comes, okay, enough of biology, now can you translate that? So that question always comes, and then we think, okay, how to tackle that, and that was some of the initiatives I will discuss with you. But as you know that I'm not a chemist, so all the synthesis and other parts are done by collaborators, and mostly our approach is looking at DNA replication process in pathogens. Here, today I'll be talking about Plasmodium falciparum. And the reason is, as you know, that during each cell division cycle, each and every organism and the cell has to replicate its DNA very precisely, because either under replication or over replication is detrimental for the cell. Now, malaria causes by Plasmodium falciparum, more than 1 million deaths takes place. Now, the problem here is there's no effective vaccine, which is 100% effective, as well as the only drug, artemisinin, the effective drug. Now, the resistance is coming up. Now, the question is why this is happening, and that to artemisinin, as you know, that's a Chinese herb. It's not a synthetic drug which you can produce. There are 17 genes required, and people are now trying to put all those genes in yeast and try to get whether they can make artemisinin. But I think we are far from the reality. Now, probably this is due to lack of knowledge of basic biology and biochemical processes. So we thought that understanding of DNA replication, how the parasite multiply, will be a good part. The reason being, if you can understand how the parasite DNA replication takes place, and if you know what are the molecules which are involved in it, maybe you can come up with some intervention processes. Now, if you look at the parasite biology, infection starts with a mosquito bite, which injects the sporozoites, which goes to the liver stage. And in liver, it multiplies tremendously. And this is called liver stage, and this is the subclinical stage. You don't see any phenotype for this. But then these porozoites, then they're ejected in the blood stage, where from one infected parasite, you can get 16 to 32 new parasites. That means from one cell, you can get 16 to 32 new parasites. There's a tremendous amount of DNA replication takes place from ring, trop, and scion, and this is the clinical stage. The fever you get is basically rupturing of the RBCs, and all of a sudden, these thousands of new merozoites come, and then again in the next phase, they go and invade the RBCs. This cycle continues, and some of them form gametocytes, and these gametocytes are taken up by mosquitoes. And this mosquito cycle goes on before ejecting again into the human, and there also at least there are 2 places where multiple rounds of DNA replication takes place. So as you can see, there are 4 stages where multiple rounds of DNA replication takes place. So our goal is to understand how this process takes place. Now if you look at what is happening is there is a nucleus initially, and then there are 2 organelles called epicoplast as well as mitochondria. Nucleus keep on dividing, and this is contrary to the normal eukaryotic cells, where each nuclear division is actually then the cytokinesis takes place. But here, nucleus keep on dividing, as well as this organelle. But imagine this is an RBC, there is a limitation, so there has to be a signal which turns it on, and then after a certain time, there has to be a signal which turns it off. So this continues. So today's talk, I will mostly talk about how the nuclear division takes place, and also the epicoplast. Now, what is epicoplast? Epicoplast is a plastid-like organelle which has been taken by endosymbiont pathway. And it's an indispensable organelle for Plasmodium falciparum involved in isoprenoid biosynthesis, heme biosynthesis, and fatty acid synthesis. Now, the beauty of this, since this is prokaryotic origin, there are a number of enzymes which are strictly present in the prokaryote, including a bacterial type gyrase is present in Plasmodium falciparum. Then, of course, if that is true, then you should be able to target that and then see whether you can kill the parasites. So our goal is to look at, for the epicoplast biology, how these enzymes work and whether you can inhibit those enzymes. For nuclear division, we want to understand that how nuclear division takes place, what are the factors involved in this, and where are the sites? Are the replication initiation sites known? And if you know, then can you inhibit DNA replication specifically for the parasites, but not for the hosts? Now, first, falciparum gyrase is a potential target. Now, in the epicoplast, there is a 35 kb genome is there, which is circular. What it does, it only codes for some of the rRNAs and tRNAs. What has been shown in 1997 that you can use ciprofloxacin, and imagine in 1997 there is no genome sequence available. But they showed that if you use ciprofloxacin, which is conventionally a gyrase inhibitor, you can kill the parasites. Not only that, also they showed that using ciprofloxacin, only the epicoplast DNA is cleaved without touching the nuclear DNA. So that suggested that there could be gyrase homologs in these parasites. Now, what is gyrase? Gyrase is a topoisomerase, and it has a lot of these functions. Particularly, gyrase function is, as you can imagine, if it's a circular DNA, either for replication or transcription, what you need to do, you need to separate these 2 strands. But when you separate these 2 strands, you are creating a positive supercoil ahead. So gyrase is the enzyme which comes, cuts, and then rewind this and then religate. Not only this, gyrase also has a very specific function. For some reason, bacterial chromosome is kept in negative supercoil form. So gyrase is the only topoisomerase which not only relieves this tension, also it introduces negative supercoiling. Now the first question we asked that whether Plasmodium falciparum, when the genome came, we said yes, there are 2 gyrase subunits, gyrase A and gyrase B, which forms a heterotetrameric complex, gyrase A2B2. Surprisingly, they have similarity with E. coli, but they also have this long N-terminal extension. What are these extensions? These extensions are basically apicoplast targeting signal. Because now if you fuse that and make a GFP parasite line, you see that gyrase is actually going to the subnuclear dots, and these dots are actually apicoplast. The reason being you can co-localize gyrase signal with ACP. ACP is acyl carrier protein, which is required for fatty acid synthesis, which also takes place in the apicoplast, suggesting that Plasmodium gyrase, indeed, it goes to the apicoplast. We also took E. coli gyrase B mutant cells, and then we complemented that with the PF gyrase B, suggesting this gyrase, which we have characterized, is a functional gyrase homolog. Now, how does it work? As I have told you, the gyrase form a gyrase A2 and B2 complex. So the DNA has to pass through this gyrase B mediated clamp, and then it passes through, and gyrase A cuts this, and then after passing this, again religation takes place. And this is the model we proposed by virtue of the fact that gyrase B of Plasmodium specifically bind to DNA. Now, if that is true, how does ciprofloxacin work? Ciprofloxacin works the way what happens is gyrase A has an active tyrosine which cleaves the DNA. But then when ciprofloxacin comes, it makes a trimeric complex of DNA, enzyme, and ciprofloxacin. Normally, what happens in the absence of ciprofloxacin, the cleavage will take place, then religation will take place. But when this trimeric complex is formed, then it is found in the cut, and it is stable, and it cannot religate. So this is the mechanism, and this is the ciprofloxacin, which is known. So now we thought that if this is true, whether we can show that gyrase blocks and also cleaves DNA. And we found that PF gyrase B indeed can do the cleavage. Now, does it cleave plasmodiums? Yes, we found that gyrase not only keeps DNA, it also kills the Plasmodium falciparum, and it's a reasonably low IC50 value. That was for gyrase A, and gyrase B also has a known inhibitor called comonomycin A, which actually goes and binds to the ATP binding site. And we found that Plasmodium gyrase B ATPase function can specifically be inhibited by comonomycin A1. Again, this is in vitro. Now, when we put that, again we found very nice result, again at the submicromolar level, the IC50. So that means both gyrase A and gyrase B can be used as target for Plasmodium falciparum. Now, how does it work? Since it is affecting the apicoplast, which is an organelle, it is not affecting the nucleus, it shows delayed death phenotype. And what is delayed death phenotype? Delayed death phenotype is, if you can see this, first cycle, one cycle is 48 hours, there's actually no effect. The parasites are fine. But only in the second, if you compare this versus this, without any drug and with the drug, there's a tremendous effect. What happens, the way it affects is the apicoplast, and since the apicoplast is another organelle, it does not work in the first cycle. So it is working in the second cycle, and it's a slow-acting drug. And that's why we believe that it has the potential of being a good drug for combinatorial therapy. And why is that? Artemisinin is a very fast-acting drug. It kills the parasites, but it also remains, there are few parasites which will survive. Now, if you have a combination of drug which is fast-acting and a slow-acting, now these drugs actually can take care of those parasites which actually bypass artemisinin. So this is what we think, that gyrase inhibitors can indeed be good combinatorial therapy for Plasmodium falciparum. Now, I talked about nuclear DNA replication. So how nuclear DNA replication takes place in the parasites? DNA replication, if you look at yeast, is initiated by a 6-protein complex called origin recognition complex, which binds to ARS consents and sequence in a sequence-specific manner, which then loads this MCM2 to 7 helicase. Of course, you have to unwind the 2 DNA, which then unwinds the DNA, and then it is taken over by all these replisome proteins, including PCNA, proliferating cell nuclear antigen, which is the processivity factor for DNA polymerase. So this is initiation and this is elongation. Therefore, in the cell, if you see PCNA, that actually designate active replication foci. When we looked at the genome, we found some of the ORC molecules are there, like ORC1, ORC2, and ORC5, and also there is PCNA. We asked this simple question that how DNA replication takes place and where DNA replication takes place. We found if you go from ring to trapezoids, from one single nucleus to multiple nucleus, as I have told you, initially both PCNA and ORC components, they're all diffused. As and when cell progresses and DNA replication starts, you see this beautiful foci. They perfectly match with each other. But as replication goes on, this foci is separated from each other. Suggesting, now remember, there is a signal which turns it on, and there is a signal which turns it off. In between, you have this beautiful foci, which are actually replication foci, and where DNA replication takes place. One protein we found, ORC1, which is there in the replication stage, but it is not there in the Sijong stage, which is the later stage. It is completely degraded, whereas the other proteins are there. So ORC1 is there, initial stage, and later stage, it is completely gone. So what we believe is happening is that if you look at, this is the cell cycle, where the ring stage is compared to the G1 cycle, or preparatory stage before DNA replication. These proteins are there. As soon as the DNA replication starts, all these proteins come together from this replication foci. DNA synthesis goes on. But remember, the parasites keep on dividing. But at that time, what happens, there is a signal, and this is a kinase, which possibly phosphorylates ORC1, and this ORC1 gets degraded. When ORC1 gets degraded, this protein, they separate from each other, DNA replication stalls. This is what we have shown in the lab with many studies, and we believe that ORC1 is the protein which turns on replication as well as which turns off replication. Now, where does DNA replication takes place? Surprisingly, we have found in the Plasmodium genome many ARS-like sequences. Now, you would say that Plasmodium is 80-85% AT-rich, so possibly ARS sequences are also AT-rich. That's why just because these are AT-rich, you are getting this. But it's not. When you take ARS-like sequences from Plasmodium and put it in yeast, replace the yeast ARS-like sequence, remarkably it works. It's not AT-rich. If you take AT-rich DNA, it doesn't work. You remove some of the base residues which form these ARS sequences, again you don't see any replication going on. So that means these ARS-like sequences which we have got has the potential to become replication origin, at least in yeast. If that is true, whether they work in Plasmodium. We looked at some of these and we found that these ARS-like sequences are present many places and PF-ORC are binding there. So now the question is whether they're binding and these ARS-like sequences are actual origins in Plasmodium. Can we target DNA replication in the parasites? We looked at this molecule, which is acriflavin, which is a mixture of proflavin and triflavin. Why we chose this molecule? Acriflavin has been shown to have potential anti-cancer activity. It is FDA approved. And before World War II, ACF was used as anti-parasitic agent. After chloroquine came, nobody looked at acriflavin. So we thought maybe this is one molecule which is already FDA approved, and let's see whether it can be a good anti-malarial agent. We found, yes, both in chloroquine sensitive as well as chloroquine resistant strain, acriflavin worked remarkably, IC50 50 nanomolar. Not only that, you can see that acriflavin in the animal model, it works dramatically as good as chloroquine, and 100% survival after 21 days Peter test in the animals, suggesting that yes, indeed, acriflavin has the potential. It has potential, but how it is working? Acriflavin is fluorescent. We found that acriflavin is taken up by the parasites. And not only the parasites, only those RBCs which has the parasites. You don't see the drug in the uninfected parasite. That means the infected parasites has a tendency to take up these drugs. Why is that? Because parasites, for taking up nutrients, they have some of this transporter which helps them to take these nutrients. And there are inhibitors available. So use some of these inhibitors. And we found that when you use those specific inhibitors, acriflavin uptake was completely diminished, suggesting that yes, acriflavin, by virtue of very vigorous uptake of the parasites through this P sac, they are going into the parasites. If they're doing so, what they're doing? We looked at the Plasmodium falciparum replication foci. I'm not sure whether you can see this, but these BCNA foci, when you add acriflavin, are completely gone, like with hydroxyurea, which is also a known DNA replication inhibitor, suggesting acriflavin is actually inhibiting DNA replication in the parasites. How it is happening? We found that we went back to the gyrase because that also has a topoisomerase activity, and it also a DNA binding protein. And we found acriflavin inhibits the ATPase activity of gyrase. Not only that, it also inhibits the activity of gyrase of supercoiling inhibition. But surprisingly, we found that inhibition of gyrase activity by SEF, it is mediated by DNA. It's not by binding to the protein. So what is happening? Number one, since this parasites has a very major uptake pathways, these molecules are specifically going into the parasites. Number two, it is binding to DNA. And when it binds to DNA, it is inhibiting the proteins which are required for DNA replication as well as for transcription, suggesting that yes, this also can be used, and our animal experiments data suggest at least till 21 days, we did not see any toxicity. It is already FDA approved. So we think that this is also another very interesting molecule which has the potential to take it to the clinic. Another drug, I'll briefly talk about this. Dr. Govindaraju from JNC, who is also present here, they are working on this TC probe, thiazole comoris. I'll just take 2 minutes, I'm done. So they found, this molecule binds to AT-rich DNA. And when they bind to AT-rich DNA, they also fluoresce. Now, if that is true, can we use this for Plasmodium falciparum? The reason being, Plasmodium falciparum is 80% AT-rich. We used that, we found in the mammalian cells as well as in the parasites, we see remarkably at very low micromolar level, this drug is going into the parasites. It is binding to parasite DNA very specifically. At that concentration, there is no effect in mammalian cells, suggesting that it is possible to design parasite-specific drug to inhibit its DNA replication. And this is the IC50 value, again at very low micromolar range, and we believe that there is a potential to improve this molecule for its better activity. So what are our plans, future plans? That gyrase can indeed be targeted by ciprofloxacin as well as carbamazepine. We should use this as a combinatorial therapy. Nuclear DNA replication, which takes place at replication foci, and ORC1 has a regulatory role. We have found these ARS-like sequences, which indeed we believe could be used as origins in Plasmodium. So we can find those ORC binding sites as possible origin sites in Plasmodium. SEF, indeed, we believe is a potent antiviral in vitro as well as in vivo, and it can be tested in combinatorial therapy. Targeting AT-rich genome of Plasmodium falciparum could be a useful strategy. This work is mostly done with all the students, mostly by Srikant, then Rahul, Abhijit, and these are the funding agencies, and these are all the collaborators, and particularly Humboldt Foundation, and thank you very much for your attention.
Department of Parasitology and Mycology, Institute, Pasteur. And she got her PhD degree from University of Delhi in the area of biomedical sciences. And she joined the International Center for Genetic Engineering and Biotechnology as postdoctoral fellow. In 2010, she got Innovative Young Biotechnology Award from DVT to conduct her independent research on molecular signal and machinery involving the egress of Plasmodium falciparum. And she was a visiting scientist in Pasteur Institute, Paris in France. Her field of expertise is parasitic infections and on molecular signal and signaling machinery. And her lecture title is shown here. Her area of expertise is again on infection of malaria parasites. May I now invite Dr. Shailja Singh to talk on this topic. This is shown here.
Thank you, Professor Chakravarti. I know you are quite strict about the timings. I will maintain the time. So first of all, I would like to thank Professor Mukesh and also Professor Devnath for this invitation, and I am really happy to be here. So today my talk is going to focus on very recent work of us, where we were trying to understand the malaria parasite biology, how the malaria parasite infect to red blood cell and how it exits from the red blood cell. And also I would like to thank Professor Dhar, because he has made my talk little easy by introducing already malaria and its life cycle. So, but to get you into my talk, I would just like to take your attention to this stage. So this is the blood stage life cycle of malaria parasite. So basically when the parasite comes from the hepatic stage, it invades to red blood cell, and then as he already discussed, after multiple cycle of replication, it forms a multinucleate shy joint, and then from again this stage which we call shy joint, this parasite comes out and again re-invade the new red blood cell. So that's the way this parasite maintains its blood stage life cycle, and all the clinical symptoms of malaria is associated with this stage. So basically in our study, we want to understand how this parasite invade to red blood cell, and how after this multiple cycle of replication and maturation, how these parasites they come out, how the parasite knows now the time has come, we are mature enough to exit and again re-invade to new red blood cell. So for that, as you see, the parasite is already enclosed inside the red blood cell by two membranes. Like one is the parasitophorous vacuolar membrane and another one is the red blood cell membrane. So to come out from this red blood cell, it needs to dismantle both the membranes, PVM as well as the red blood cell membrane, and then it comes out. So the previous studies, they discuss how this parasite they come out, how it dismantles the red blood cell or the PVM. But the question which we wanted to understand, what is the signal? How the parasite knows now the maturity has come, I need to come out. And for that, as we all know, that intracellular calcium is the ubiquitous signal, and it's a signal for life and death. So we also wanted to explore whether this intracellular calcium in our parasite is the signal for exit or not. So the simple approach we took is, we wanted to just see the level of intracellular calcium at the time of parasite exit. So here is our red blood cell, normal, and this red blood cell is infected with the parasite, and we have loaded this parasite with calcium sensitive dye, so we can monitor how the calcium flux is going to be during the parasite exit. So I am going to run a live cell movie. You are going to see the parasites are going to come out from this red blood cell, and you will see a calcium spike. So in this, you can see here is the egress process. Now the parasites are out. So just before the egress, there was a flux in the intracellular calcium. So here is just a snapshot to again make my point that yes, there is an increase in intracellular calcium before the egress of malaria parasite. So the question is whether this increase in intracellular calcium is really required for egress or not. So the simple approach we took is we chelated the intracellular calcium of the parasite as well as we chelated the outside intracellular calcium, outside calcium of the parasite, and then asked what happens to the exit process or the egress process. So here you can see when we chelate the extracellular calcium by EGTA, we don't see any change in the egress process. However, when we chelate the intracellular calcium of the parasite, we don't see egress. So at the same time, when we were doing for the calcium experiment, we also wanted to see what happens to the red blood cell membrane, whether just before egress, this host cell membrane gets permeabilized or not. So you can see the movie again, where here we are just probing our red blood cell membrane, and here in the media we have put a dye, and after permeabilization of the membrane only this dye enters. So whenever you will see a signal on top of this parasite, like the surrounding of the parasite, that indicates now the host cell membrane is permeabilized. So here you can see, and here is some volume and the diameter measurement, and you can see here just before egress you can see the green signal which is around the parasite, which represents now the host cell membrane is permeabilized, and after this permeabilization process the parasites now they are coming out. So again I have a snapshot for this. So you can see now the parasite is enclosed with the red blood cell membrane, but it's not permeabilized, and before the egress of the parasite the membrane gets permeabilized. So the question is whether this permeabilization of the host cell membrane is also calcium dependent. If we chelate the calcium, what happens to host cell membrane permeabilization? Because the host cell membrane permeabilization is really required for the egress process. So again, when we treated our parasite with the BAPTA AM, which chelates the intracellular calcium, we don't see the host cell membrane permeabilization, and also we don't see the egress process. Now the question was, what is making this host cell membrane permeabilization? So for that, we were looking for some of the pore-forming proteins in the parasite, and we came across with these perforin-like proteins. They are expressed in the parasite, and there are 5 perforin-like proteins already annotated in the Plasmodium genome database. However, when we made the specific antibodies of all these perforin-like protein, we could see only perforin-like protein 1 and perforin-like protein 2 are expressed in the malaria parasite, and then only perforin-like protein 1 is on the host cell membrane. So our observation says that our host cell membrane gets permeabilized, and then with this information we can see, yes, there is one pore forming protein expresses in the malaria parasite. At the time of egress, at the very late stage, they are present in the host cell membrane. So and then we again probed, because if this protein is already present in the host cell membrane, it's not, we are going to get the immature permeabilization and the egress process. So to see whether they are present in the specific location of the parasite, and when they get signal, it's been discharged to the host cell membrane. We did this localization with the probing of some organelle specific protein as well as this perforin-like protein. So no need to go to very much detail, but here you can see in green is inside the parasite, and here red is the host cell membrane. But when the progression happens and the parasite matures, this green protein representing perforin-like protein, they come to the host cell membrane, and then this is the free merozoites. So also, as I said, these parasites are enclosed by 2 membranes. We also probed whether this other membrane also contains this perforin-like protein in this late Shailja stage or not, and we could see yes, even in PVM, which is a other limiting membrane, it is also present. But when we chelated the intracellular calcium of the parasite, this protein is not in the host cell membrane. So ultimately, this leads to the conclusion that parasite expresses a perforin-like protein, and when the parasite calcium goes high at the time of egress, it leads to discharge of this perforin-like protein, and that discharge leads to its presence in the host cell membrane, making the host cell membrane porated, and that's what it acquires some ion flux and the media from outside, and then parasite exit from the red blood cell. But the question is, every time I am saying the intracellular calcium is required for this and that and also for egress process, how this intracellular calcium gets inside the parasite. What is the real signal, endogenous one, which really triggers this intracellular calcium? And for that, we look into the literature and we know that the abscisic acid, which is a plant-like hormone, it's a second messenger, and also it controls the release of intracellular calcium by CADPR. And some of the epicomplexins also already been shown that CADPR, it controls the intracellular calcium. And one of the epicomplexin, similar like Toxoplasma gondii, which is again a parasite, it also expresses abscisic acid. So our question was whether our parasite also expresses the abscisic acid, which is a plant-like hormone. So the next thing was, again in the literature, some of the precursors were already reported, but not the abscisic acid. Like for the synthesis of abscisic acid pathway, you will find there's an enzyme phytoindesaturase, which converts phyto into all trans-phytofluene. So here, this is an important enzyme, and also some of the precursors is already been reported in the parasite, but the abscisic acid was not. So we used one chemical, it's a herbicide, fluridone, and it's been known to inhibit the phytoindesaturase. So when you inhibit this enzyme, you don't get the abscisic acid. It's been reported in the plant. So first thing was, we asked whether the abscisic acid is there in the parasite or not. So we made the lysate, we did the LCMS analysis, and here is the data. We could detect the plant-like hormone in our parasite, and that too in a time-dependent manner, because this abscisic acid is not expressed in the early stage. This hormone is expressed very late stage when it is required, and that's what we see a very change in the intracellular calcium at the late stage, 48 hours. Maybe that threshold is needed, that abscisic acid threshold is needed for this calcium increase at this point. And then when we treat our parasite with the fluridone, which blocks the phytoindesaturase, we don't see abscisic acid. And when we don't see abscisic acid, we also don't see increase in calcium. So everything is falling in line in sense that our parasite expresses the abscisic acid, and when we try to disrupt the abscisic acid synthesis pathway by using the fluridone, we don't see abscisic acid, and then we don't see increase in calcium. And also, if we don't see increase in calcium, we don't see egress process, like the parasite are not able to exit from the red blood cell. But when we provide the abscisic acid from outside, we do see the egress process. That again shows that as a specific process, the abscisic acid is really required to increase the intracellular calcium at a specific time for the egress of the parasite. So as I discussed in my previous slides, that yes, permeabilization of the host cell membrane is really required. So in fluridone treated Shailja, where the abscisic acid is not present, our parasite host cell membrane is not permeabilized, and also the perforin-like protein is not on the membrane. And we also proved this by Weston. So the story so far, which we want to say that maybe this abscisic acid pathway do exist in the parasite, and because of the increase in abscisic acid at very, very late stage, it influences the parasite intracellular calcium, and this intracellular calcium lead to discharge of pore-forming proteins as well as some of the proteases, which we have already shown in our previous work, and that leads to egress. So finally, and that's what we claim, that this is a very specific to ABA synthesis pathway, is very specific to parasite. In human, we don't have that pathway, so it's a good target. And if at all this herbicide already I have seen in the literature, some of the treatment of parasite is already been used, but in malaria we are trying to use it for our animal model, Plasmodium burgii, and it is really showing a good progress. But we have to check for other like the necessary phyto, like its property, how it's been secreted and other thing. So I was funded with the Department of Biotechnology, as Professor Chakravarti has already told about that, and these are my students, Swati Garg, Shalini Agrawal and Manish. They are also DBT, JRF and SRF. Poonam Dangi and Vijeta Sharma, they were also involved in they are now involved in the animal work. They are the student, and Shonal Gupta is a postdoctoral fellow from UGC working in my group. And previously, I started my parasite work with Dr. Chetan Chitnis lab at ICGB. There we also worked on signaling process in invasion of malaria parasite. So I'm happy to take any question, and I think I maintained the time. So basically here yeah. Yes. So basically, for our normal movie, when we do the time-lapse experiments, we do see the increase in calcium flux, which is very, very high just before the egress process. But if you measure the total intracellular calcium with different time point, even it starts from 42 hours to 48 hours. So it's somehow it builds up the calcium level at the time during the maturation, and just before egress, maybe because what we do see is a flux, maybe because of the permeabilization, now the influx of even from outside. But the abscisic acid mediated calcium is maybe at 44, or because we cannot measure the abscisic acid in our time lapse movies. We don't have these indicators. Yeah?
How long does it take for intracellular calcium to go up above an hour?
Okay, so it takes 20 minutes. 20 minutes? Yeah. When we provide from endogenous abscisic acid to revert back our egress for our egress of parasite, we take the stuck Shijones, we provide the abscisic acid, we wait for 20 minutes and then check for egress process. And then in our live cell movie, when we put the abscisic acid in 5 minutes, we do see an increase in calcium. Okay. Okay, so I didn't showed you that data. It's not the calcium-induced calcium release. It is basically maybe through CA-DPR pathway. It's calcium release from the ER through ryanodine receptor. Because when we block the so that's the abscisic acid. So that's even in plant it's not clear. So abscisic acid builds up, and then for some unknown mechanism it also increase the CA-DPR level, and CA-DPR is already been known for inducing calcium from ER. But that part even in plants is not clear, that how this CA-DPR from abscisic acid to CA-DPR, and for that maybe some receptors are involved. You are right, but in Plasmodium nothing is known, and that's our next project, which is that receptor in the parasite. Maybe that will be also a very, very good target if you can find it. Yeah. Yeah.
Very interesting concept, which shortly I touched it in the morning. I have two major issues now with this concept, both in the plants and now in the Plasmodium. If this acetic acid is really made intracellularly, then we have a very different concept of signaling, because all classical hormones are coming from outside, not coming from inside. So you have now to postulate that there is an intracellular whatever receptor or structure or signaling structure. So that might explain your delay, because when abscisic acid is only acting in the cytosol, typically where it's synthesized, it is not like an autocrine mechanism.
Yeah.
What is your hypothesis there?
Yes. So when you see the plant side, the abscisic acid signaling is internal. So the plant even produces the abscisic acid inside the cell, and the receptor for abscisic acid is sitting in the cytosol, and that is linked with the guanyl cyclases, and then you get the cGMP level high. That leads to somehow release of calcium in plants. But in some cases, it's also been shown that abscisic acid from outside also, some receptors are present on the membrane. On the cell surface. But my hypothesis for the parasite, I expect maybe the receptor is like the first one, the cytosolic, and that's what we are very close to that story also. I have found G protein coupled like receptor, cytosolic, which is very vesiculated and it has a capacity to bind abscisic acid. But as it's very tough to create a gene knockout in the parasite. Now the technology is not that advanced, but now I'm in Pasteur and we have now very new CRISPR system has come where I can make a very good gene knockout. So we are going to try this protein coupled receptor, which we think is an abscisic acid receptor, and then we'll simply ask a question: what happens to parasite?
When you look at the absolute quantity of this abscisic acid on mass spec, you only focus to the Shailja and merozoite, not.
No. We like you can see my slide. We measured the abscisic acid in all the stages. So here is the data. In red blood cell, we don't see abscisic acid, but when we take the 10-hour rings, there's some level, and 34s, 48. So we do see a significant amount of abscisic acid in 48 hours Shailjas and the merozoites. So, and that makes this story more strong, that if the abscisic acid is throughout high, how are we going to give that hypothesis that the calcium is really high at the late stage controlling the egress process?
Another thing is that I'm trying to correlate the levels of the calcium in the egress process with the membrane deformability. Because you mentioned that the absolute rise in the calcium level is prerequisite for the egress.
Slide for that.
This one?
Yeah.
So here is like in normal mature Shailjas, you see a protein which is a pore forming protein, it's on the membrane. But when we treat with the fluoridone, so there we are just inhibiting the abscisic acid formation. So ultimately the calcium in surge. And then you cannot see this perforin-like protein now on the membrane of the host cell membrane. And when we analyze these kind of parasites, they are not even permeabilized. You can see here there is no red staining which is coming from the phyloidine, and this you always see when the cells are permeabilized. So these cells which are treated with the fluoridone, they are not permeabilized, and here we have put the dextran. Again, dextran will enter when the membrane is permeabilized, and here you can see the dextran is very well entered here but not here.
So does it mean that fluorine also has an effect on the perforin trafficking as well?
Yeah, because the first observation was the intracellular calcium controls the egress because it regulates the perforin discharge. But what controls the intracellular calcium is the abscisic acid. So here from this story many targets are coming up. Like you can target the pathway for abscisic acid, you can even target perforin-like protein, which is again we are doing we are taking a chemical biology approach. We have now some drugs which really binds to this perforin-like protein and they inhibit their action. And there also we are not seeing the permeabilization and egress process.